Thank you very much. Thank you very much for joining us this afternoon. I see some familiar names there. I see some serious neuroscientists there. It's terrific to have such a mixed audience attend this afternoon. So just for those who don't know about the WPC, you should know that the next major WPC conference is in Arizona in 2026, in Phoenix, Arizona. And it's been going since 2006. And as Eli said, what we try and do is we try and connect all the different stakeholders in a unique way. So we try to bring scientists, clinicians, therapists, patients, caregivers, everybody together. And so this research spotlight, which has now been running for a number of years, is a way in which we put someone under the spotlight. So Philippe, Huot, I don't know if I said that correctly, is our guest today. And what we like to do is I like to find out a little bit about Philippe, what makes him tick, where he was born, how he ended up in Parkinson's disease. Then we're going to talk about his area of expertise and research, which will be around involuntary movements induced by medication, so dyskinesias. We're going to discuss a little bit about that. And what's great is if you can just keep your questions coming. So if you do that, put them in the chat, and I will endeavor to keep an eye on the chat so I can bring those questions as and when they're relevant. I'd also like to thank some of our previous research spotlight participants who I noticed are also on this call listening in. So Philippe, perhaps it would be useful for you just to say who you are and where you are currently. What is your current position? And then we can go back a little bit in time to where you began life.
Sure. Well, thank you very much for having me. Actually, it's a real pleasure to be here. So I'm an associate professor of neurology and neurosurgery at McGill University in Montreal. I'm scientist at the Montreal Neurological Hospital and Institute, and I'm also a movement disorder neurologist at the McGill University Health Center. So I do research, and I also see patients with Parkinson's.
Okay. So you are that unique beast of someone who spends time in the clinic and time in the lab.
Am I really a beast? Yeah.
Okay, so Philippe, I mean, it's useful to know where you sort of began. So where did you grow up? You know, how did you get into medicine? Tell us a little bit about that.
Yeah, so actually, I was born in Quebec City,
Quebec.
In Canada. And I mean, how did I get into medicine? It's actually a good question. I've always been interested by the brain and how it works. And I guess I've always been curious about if it works well and what happens when it doesn't work well. So that would be like in diseases. So then I was thinking, how could I just learn more about the brain? And there were like many options, but one which I thought was very interesting was medicine, because we get to see brain conditions. We get to see a bunch of people who are healthy, who have brain conditions, which is why when I entered medical school, I was kind of decided to go either in psychiatry or neurology, and I ended up going into neurology in the end. Yeah.
Okay. So you grew up in Quebec, which is a beautiful city, Quebec City. So very French, very beautiful.
Yes.
And did you go to medical school in Quebec, or did you go to Montreal, or did you go?
So I actually did my medical school in Quebec City. Yeah. And I also did my master's in Quebec City as well, before moving on to Toronto to do my PhD.
Okay. So, and when you were doing your sort of medical degree, you already knew whilst you were studying medicine as a medical student that it was brain, and it was either psychiatry or neurology. So it wasn't as though you're interested in the gut, or you knew north of the neck was where you were going.
Yes, exactly. Not south. Yeah, definitely.
And so during that time, you obviously then trained as a doctor. And then in the Canadian system, how do you have to do a lot of general medicine before you go off and train as a neurologist, or did you just go straight off and do neurology training and then a PhD?
So first we need to do medical school, which depending on how long is it is either 2 or 3 years. Yeah. Then we need to do clerkship, which is 2 years, where we kind of touch upon everything. And then we enter residency, which depending on the selected residency is between 2 for family medicine to 5 years for neurology and 6 years for cardiology and neurosurgery. What's interesting is that, so the brain disciplines, I'm going to call them, is that you get to select them after med school, after your clerkship. So I applied to get into neurology, and I was fortunate to get into neurology and to become a neurologist.
Okay.
So that's how it works in the Canadian system, essentially.
So you're a bit unusual, I would say, in the world of medicine, because most people are a little unformed when they start, and then they end up with their specialty having had a little exposure, whereas you were quite clear where you wanted to go. And so you chose neurology over psychiatry, and then you went to Toronto to do your research. So that was, you'd already done a bit of neurology training, and then you wanted to specialize and do research.
Yeah, but I kind of have to tell you, so my exposure to research in Parkinson's disease was a bit of an accident. So in Quebec City, you're probably familiar with Dr. André Parent. So he's the author of the Carpenter's Human Neuroanatomy. Because at the time, I was interested in spinal cord. And I was also interested in doing research just because in medicine, we treat patients, but I felt that we don't get to the bottom of why diseases happen and why treatments work the way they work, which is why I ended up contacting Dr. Parent just to inquire about possibilities and to have a chat with him about his book, all that. And then I continued. So I did a master's with him in neuroanatomy of the basal ganglia. And this is really where I started liking.
So these are the structures deep in the brain which are intimately associated with the pathology of Parkinson's.
Completely. Yeah, that's where it goes wrong in Parkinson's to some extent. And then, I mean, I was like, during my residency, I tried to do a bit more movement disorders after that, because, I mean, that's what I did with Dr. Parent. So I was exposed to pathologies and conditions like Parkinson's. I saw a couple of patients with Huntington's. And this is where really I decided to specialize more into movement disorder. And I knew that they had a very strong group in Toronto. So I ended up contacting Dr. Jonathan Brocci at the Toronto Western Hospital. And I mean, I just want to say that I've been so lucky to have incredible mentors, Dr. Parent, Dr. Brocci, Dr. Susan Fox as well, Dr. Lang. I mean, it's like, this was just an amazing place to learn about movement disorders in terms of clinical aspects, but also preclinical aspects. How does thing work? What is the pharmacology of the movement disorders and the basal ganglia? That was just amazing. So that was essentially my training in movement disorders.
Well, it was very good of you to work with Dr. Brocci and Susan Fox, because they're British, and you're from Quebec City. So Quebec, so the French of Quebec, and the British of the UK have had a long history, not necessarily of collaborating, but that's a bit in the past. So I just want to come back to this master's which you did with André Parent about the anatomy of the basic. I mean, what was the main bit of that work? Was that about just understanding in the anatomy, or were you looking for pathology in a particular condition?
So we studied, and at the time, to me, I was not aware that they existed. So in Parkinson's disease, we talk about dopamine all the time, right? So there's a part of the brain called the substantia nigra that produces dopamine, and that ends up, for some reasons, degenerating. There's less dopamine in another brain area called the striatum. So we worked on that brain area, the striatum, because Dr. Parent, a few years before, had discovered that there was, Dr. Parent and the work of others as well, they had discovered that there are some neurons inside the striatum that produce dopamine. And our question was, what is happening to these neurons in Parkinson's disease? which is a condition where there's less dopamine, and what happens in another condition where there's more dopamine, which is Huntington's disease. And then we tried to discover in another project what is the influence of dopamine replacement on the number of these neurons? Because what we found at the time was that the number of neurons do tend to go up in Parkinson's, maybe as a compensation mechanism, albeit imperfect because people are Parkinsonian. And the L-Dopa treatment reduces the, kind of reduces this compensation. So essentially, it's probably a mechanism that the brain has to compensate, but it's limited because it doesn't produce enough dopamine, and there may not be enough of these neurons in humans. But I thought it was just like absolutely fascinating.
Yeah, because I remember this quite clearly. So basically, as the dopamine innervation of the striatum disappears, so these new population of cells appear in the striatum. Well, I don't know if they're new population, but there are cells that start making dopamine. But obviously, they don't do it to the efficiency where they can compensate for the lost dopamine. Otherwise, you wouldn't have Parkinson's in the first place. So, I mean, I want to come back to that because I'll be interested to know whether you think that feeds into dyskinesias or has some role in that. But obviously, that was a sort of fairly formative period in that lab, and obviously very associated with Parkinson's. So, when you went to Toronto, what was your research project there? What was your PhD? Did you have a clear idea what you wanted to do, or was it more that you arrived and they said, Philippe, solve this problem for us?
That was kind of the latter, I would say. So I mean, I remember my first meeting with Dr. Brocci in Toronto. So he had a piece of paper and he had written 3 words. So he said, so this is going to be your PhD. And I mean, that was very interesting, but at the same time, that was just an amazing project because there was a lot of creativity allowed. And of course, a lot of like tools and resources were provided in this like state-of-the-art environment. And I think this is important as well because, you know, after you finish your studies, if you want to be a PI, I mean, you kind of need to be able to think on your own and to design experiments and to be able to answer those questions. So yeah, but I had a general idea of what I wanted to do, not necessarily this project in particular, but I wanted to learn about the pharmacology of Parkinson's disease. How do dyskinesias and hallucinations, because I also worked a lot on hallucinations and I still do, how do they happen? And as a physician, how can we alleviate these manifestations? So this is what I worked on. This is what I'm still working on to this day, essentially.
So it's a very clinically orientated problem. It was, you know, one of the problems with Parkinson's, especially its treatment, is the evolution and development of these L-Dopa-induced dyskinesias, the neuropsychiatric problems with hallucinations. So that is a very sort of, and you're absolutely right. I mean, as you will know as a clinician, these are still major problems in the clinic today. But you were trying to solve the problem as to what drives them, and if we know what drives them, how we can better treat them, which I would say is a little limited even today. So what was the PhD on? So how did you set about doing that? Because these, you know, someone who's you know, not necessarily in the field, they'll say, well, these, I mean, how do you know if you're going to model this in the lab whether the animal or the cells are hallucinating? I mean, how do I know that? Because that's something which people tell you. And dyskinesias, you know, they come after years of people have taken medication in the context of Parkinson's. So how? I mean, that doesn't happen in the lab. So no animal, as we've discussed before on these research trials, gets Parkinson's apart from us humans. So how did you set about trying to solve these problems? Because the first problem you've got is there's nothing in the lab that looks like it. Looks like what you're seeing in the clinic.
I mean, it's actually a good point, what you're saying. And I mean, I think we need to take animal models for what they are. They are models. And I mean, if we look in the clinic, when do people have dyskinesia? So there's a few things that they have in common. I mean, first, they've had Parkinson's disease for a certain number of years, although this is not an absolute. they've been exposed to dopamine replacement therapy for a certain time, which again is variable. But from these two items, it's kind of what we're trying to model in preclinical settings and in experimental models of Parkinson's disease. So we need to have a Parkinsonian state, which in experimental conditions we define as we said before, dopamine reduction in a brain area called the striatum. Okay. And then knowing that dyskinesia is triggered in the clinic, or at least elicited after treatment with levodopa, we expose our preclinical models to levodopa on a daily, regular basis until they do develop dyskinesias, which we then, and I mean, then what we do once we have generated a dyskinetic phenotype is essentially limitless, depending on the models that we're using and the research questions that we want to answer.
Okay. So the fundamental model is, you say dyskinesias happen in clinic as a function of Parkinson's disease and the dopamine loss and dopamine replacement. Therefore, modeling it means removing dopamine in an animal model, so lesioning the dopamine nigrostriatal dopamine pathway and then exposing the NML12-DOPA. And I guess if that is correct, what you're saying is the cause in the clinic, you would expect dyskinesias to emerge, and that's what you see in the lab. So that is a faithful model of that, although it's obviously what some people say is a rather reductionist model because it only reduces it down to two elements, but they do seem to be the critical ones. And so if you do that in an animal model, how do you know the animal's dyskinetic? Do you just look at it and you can see these funny movements, or I mean, how do you sort of quantify that, if you like? How do you sort of, because obviously what we're going to come on is, what's the basis for that and how do you treat it? So how do you measure dyskinesias in animals? How do you sort of score it? Or
yeah, so I mean, I will say it depends on the animal model that we're employing. Historically, there has been dyskinesia research in preclinical models with rodents and non-human primates. I'm going to start with the rodent model first. So it's possible to elicit dyskinesias upon causing dopamine reduction in the striatum in both mice and rats. Okay. And historically, some people in the attendance may know that as well. So the measure that we were looking at was like in rodents, because we lesion one side, is the number of rotations that the animals were doing. And this has been the standard until the late 1990s, when there were a few seminal studies published by the group of Dr. Cenci in Sweden that essentially invented a scale to rate dyskinesias in rodents, both rats and mice. And it's called the Abnormal Involuntary Movement Scales. And there are some parameters that we look at. So we look at, for instance, the orofacial movements, the movements of one limb, also the twisting of the animals. And it can be scored based on the duration and also based on the amplitude of these movements. This has really standardized research in dyskinetic rodents across different labs across the world, and it's now employed worldwide to study the effect of drugs on the severity of dyskinesias and also to study the mechanisms whereby dyskinesias may happen.
So can I just also, so I mean, this is great because obviously now you have a standardized score. So what you do in your lab, Angela Cenci in Lund does the same, groups in it. So everyone's scoring it the same way. So two things I just want to ask. First of all, so obviously when the rats rotate, which is the standard model, you do a very complete lesion on one side of the brain. In the dyskinetic models, do you do lesions? Do you knock out the dopamine system on both sides, or do you just take it out on one side and that gives you the dyskinesias? That's the first question. And the second is, how automated is this? So can you now just sort of stick a camera on this cage and record it, or does it still rely on you sitting there scribbling in your little book about about what these animals are doing?
Yeah, so the first one is that, so the first question, which is unilateral, bilateral rotation. So, I mean, essentially, it's bilateral lesions and injection. Those models are produced with 6-hydroxydopamine in the ventricles are not done as often as they used to be done these nowadays. Because the animals become non-motivated and extremely Parkinsonian at the beginning. And quite frankly, a lot of the questions can be answered by performing lesions on one side of the body. So
your standard model will be a unilateral one-sided lesion.
Exactly.
And then you give them the L-Dopa and you can see the dyskinesias in that.
Yes.
Okay. So that's
And
useful to know. And then the automation of that, is that now, can you use some sort of computer system and video system to score it all in a more objective way than you looking at it, or is that still not really viable?
it still relies, I mean, I will say I'm quite happy about that. It still relies on art and the rater experience and the use of the scale, which introduces the possibility that there might be a bias.
Yeah.
But the thing is that if the studies are done in a randomized fashion, and if the person injecting the treatments, for instance, and the raters are blinded to the treatment, theoretically, this potential bias could be eliminated. And this is what we try to do when running studies in the rodent, because I mean, the possibility of the self-fulfilling prophecy for efficacy or non-efficacy of drugs is real in the case that a rater wouldn't be blinded. So this is kind of a safety mechanism. I know that there are some detection systems being developed, and I mean, I do think that at some point these studies will rely less and less on human raters.
Yeah.
But I kind of like it that there's still art and research as well.
Okay. And I interrupted you. I'm sorry about that. Because obviously you mentioned non-human primates, and obviously this is another somewhat contentious area of research. Should we do any research in that? But I mean, obviously there is a history that people have done work in this area. So, I mean, I'm just interested to explore. Do you think they give us additional information? Is there, that approach which you've used in the rats? Obviously, you could do similar approaches in non-human primates, monkeys. Does that give us better information, different information? How valuable are those type of models for this type of work? And also, if you were to embark on that, if someone was interested in doing that, what's the sort of a process? I mean, presumably it's very highly regulated.
Yes, I mean, maybe we could talk about the regulation first, and then we could talk about non-human primate. I'm actually a member of the ethics committee at the Montreal Neurological Institute, and animal research is actually regulated in a different numbers of ways. The first one is, let's say I get a grant. So for a specific project that has been scientifically peer reviewed, I will not be able to use that grant money before the project has received approval from the ethics committee. Okay. So this is the first step. And second is the composition of the ethics committee. So
it's just like a clinical trial. I can get money for a clinical trial, but I couldn't start a clinical trial unless I had ethical permission, permission from an ethical committee to do it. Okay, so it's exactly the same with animal work.
Absolutely. And once the scientific merit of the project has been assessed, then there's the ethical part and the animal welfare part of the project that is assessed by the ethics committee. At the Montreal Neurological Institute, there are 2 vets on the ethics committee. There are students, there are principal investigators, there's a community member, there are animal health technician, and there's a representative of McGill University Compliance Office. So in addition to the scientific merit, your project must be evaluated for safety, animal welfare, And only then, if it fits all these boxes, will the project be approved, and only then will you be allowed to start working with animals. So it's highly regulated. And I think it's important because we as researchers have a responsibility. We want to make the human condition better, but we also want to respect and take care of animal welfare. And every year when we renew our ethics protocols, so they're renewed on a yearly basis, we need to say if we have made improvements in the past year on animal welfare issues. So it's like, so it is monitored, and I think this kind of oversight is very important.
Yeah. I think I'm very grateful for you to talk about that, Philippe, because I think it's something we sometimes slightly gloss over, and I think people who aren't involved with research may have a slightly different view on the fact that these experiments just go ahead without much thought. And in fact, there's huge amounts of thought and care that goes in to all preclinical work to ensure the welfare of the animals, and that the experiments being done have merit and clearly have relevance to what we're looking for in the clinic. So coming back to the non-human primate, there are studies which have been done in that, and I'm just interested to know in general, has that been a big boost to the field of dyskinesia research, which is where I want to go next, because I'd quite like to know what causes dyskinesias and what you're going to do to help me treat my patients with it.
I would say that without non-human primates and without rodents, a bunch of treatments that are either being tested or have been approved would not have made it through to to clinical testing and approval. I'll give you an example. I mean, for dyskinesias, for instance, we don't have much to offer to patients, right? I mean, we have amantadine and maybe clozapine that we can talk about later. But amantadine was shown to be efficacious in rodents, and was also shown to be efficacious as well in non-human primates. But if it hadn't been the case, and that's the question, would the development of amantadine have been pursued? If there's no efficacy in animal models, is it worth exposing humans to an experimental molecule that can have no efficacy and adverse effects? Because that's also something that those preclinical models, and maybe on that I will say especially the primate are important, is that when you do preclinical research with a molecule, you're just not looking at the efficacy of that molecule, but you're also looking at, okay, this molecule has an effect on dyskinesias, but what are the levels in the plasma?
Yeah.
Because if sometimes the efficacy for dyskinesias is at plasma levels X in an animal model, and then you go to the clinic, and when you reach plasma levels much lower, and it's poorly tolerated by humans, then you're going to say, well, we're probably not going to be able to reach those plasma levels. So that drug may not be well tolerated by people. So it's probably not worth pursuing it to risk eliciting adverse effects and in people. So there's also that's very important. So it's not only efficacy, but it's also safety to humans, because we don't want to embark or administer a molecule to people if we know it can harm them. So the animals, especially, very helpful. And the other thing that an animal, either a primate or a rat, is very helpful is, I know we're trying to do a lot of stuff in silico and in cell models, but at some point, we need to have a complex organism that has a liver that will metabolize drugs into metabolites. What's the effect of these? What's the global effect? Does it enter the brain? Even if it's modeled too, is it metabolized too quickly in either a rat or a primate? And then the compound that we're trying to get to the brain gets metabolized, and then we have no effect. So these are all questions that have to be answered before moving to humans. Otherwise, there's a safety issue for humans and a possible lack of efficacy.
So basically, you need the models in order to not only show that your therapies work against what you're dealing with, which in this case is dyskinesias, that they also don't have a whole series of other effects, and that the doses you're ultimately going to aspire to use are within a reasonable sort of level to be tolerated. So you've got your models, and you've been talking about the fact that clearly two critical factors are the loss of the dopamine pathway and then the dopamine itself, L-Dopa. So one of the questions is, there's a question about mucuna is less dyskinetic than L-Dopa, but I want to sort of broaden it out and say, you've talked about lesion and you've talked about L-Dopa, but in the clinic, what would be the critical factors determining when people get dyskinesia? So what would be the sort of factors which, because we'll have people with Parkinson's on this call, what would be the sort of, if you were seeing people in clinic and they said, I'm worried, because I have this a lot with younger patients, for example, they won't take L-Dopa, they won't take Sinemet or Madapar or equivalent in different parts of the world because they worry about the dyskinesias. So what would you say to those young patients, and what would you say are the critical factors determining whether someone gets a dyskinesia?
I think I've had this conversation like a couple of days ago with a patient. I mean, it's like, it's true. I mean, I always like to go back to the Sydney multi-center study for Parkinson's disease. I mean, at some point, most patients will get dyskinesias. I mean, dyskinesias are something that may or not happen, because the fact that most patients get dyskinesias doesn't mean that they will all get dyskinesias. And it doesn't mean that dyskinesias are going to be interfering with people's quality of life. And I do find that this quality of life aspect is often neglected for what may happen in the future. I mean, because patients, if they are stiff, if they have pain, if they have tremor, if they're young people with Parkinson's and they can't play with their kids, for instance, maybe a treatment could be envisioned for good quality of life. And if the dyskinesias happen later, then maybe we can try to deal with that. But people with young onset Parkinson's disease, at some point, may be more likely to get dyskinesias, because if they get Parkinson's at a young age, they'll have Parkinson's for many years, and will be exposed to L-Dopa, Levodopa for many years, which increases the odds of developing dyskinesias. Yeah. But I think that those people must remain hopeful. Yeah. Because that's what I tell them sometimes.
Yeah, I mean, I often say it's a function of disease duration and dose of medication. So the sweet spot is giving people enough of the medication, but not too much. But often, one of the problems is that people sort of undertreat themselves, exactly as you say, Philippe, and they deprive themselves of the benefits now for the worries of tomorrow. And I suppose where we're going to go next with you is going to ask you about what is tomorrow and the treatment of dyskinesias. And I think there was a very interesting study, the L-Dopa study done 20 odd years ago, where they took de novo people with Parkinson's and gave them different doses of dyskinesias. And even new onset patients, if you give them very high doses, you get dyskinesias. And if you wait a very long time and give a modest dose, you can get them. So it's clearly stage of disease and dose of medication. So why do people get dyskinesias? What is the basis for it? What causes them? And then you've talked a little bit about amantadine. So I'd be interested to know, if you see people with dyskinesias which are troublesome and getting in the way, how you would treat them. So what causes L-Dopa-induced dyskinesias? And what's the current treatment for them, and then we can come on and discuss how can we do that better going forward.
Yeah, absolutely. So you and I, so people without Parkinson's disease, we have constant dopamine levels inside the brain because of what's called the dopamine transporter buffering effect, meaning that the dopamine that we produce in our brain is released by dopamine neurons that have all what it takes to regulate and to release dopamine in a physiological manner. At some point, so early on, when we administer levocarb or levodopa to patients, they're doing very well for a while with only 3 doses a day. And this is called the honeymoon period because there are still some remaining dopamine terminals in the striatum containing the dopamine transporter to release it in a physiological manner. But as the disease progresses, levodopa still keeps its anti-Parkinsonian action, yet there are less and less dopamine terminals, meaning that it's converted into dopamine by another system. And there's different systems that can do that, but an important one is the serotonin system, which has everything it takes from the proteins to the enzymes, all that, to convert levodopa into dopamine and to release it. The problem is that it doesn't have like the mechanisms to release it in a manner that is physiological, meaning that
they can't take it up either. So once it's gone, I can't get it back.
Yes. So whereas you and I have regulated levels of dopamine, people with Parkinson's become more and more reliant and depending on every intake of levodopa. And this pulsatile stimulation of dopamine receptors is thought to be an important factor leading to the development of dyskinesias. And at some point, it gets more and more difficult to separate an anti-Parkinsonian benefit of L-Dopa with the presence of dyskinesia. So for some patients, when they've had, but you realize that to have this, and this going back to what you said, that to have this pulsatile release of L-Dopa, one needs to have years of disease progression, which is why it's important to not compromise today's quality of life for tomorrow's hypothetical, because the disease will keep progressing until we have a molecule or a cure that can slow the progression. And then, I mean...
So in theory, you could not get dyskinesias if you could continuously deliver dopamine at a steady state, which of course you can't do with tablets because you'll absorb it, different absorption across the gut into the brain and such like. But, there's all these new pump therapies. So in theory, a pump therapy from the word go, if you could afford it, but I don't know what they cost in Canada, but certainly in the UK, they're not available. If you gave continuous dopamine, would you not get dyskinesias, or would you still, you think, even with an infusion peripherally, same level all the time, they would still emerge? Because you just don't have the control that the normal intact system has.
That's a very interesting point. I do believe that at some point they would have dyskinesias. And let me explain you why. So there was a study done in the 1970s by Tom Chase, where he administered intravenous L-Dopa, intravenous. So, I mean, with that, you can keep constant levels. And what he realized is that patients still had motor fluctuations. So we're not talking about dyskinesias here, but we're talking about fluctuations just because when people eat a meal and there's protein, the proteins compete with L-Dopa to enter the brain. So I'm not sure if we're going to be able to have a constant dopamine or continuous dopamine stimulation that would be even all the time to prevent dyskinesias, but I do think that we could probably minimize their severity and maybe attenuate the process leading to dyskinesia. So we may not be able to prevent it, but we may be able to delay it. But this is hypothetical.
So now you said we've got this pulsatile release, and over time, this then leads to getting dyskinesias. And then we've obviously talked about pump therapies, so you could try and control them, I guess, by changing the dose of oral medication, trying to make it more continuous. You could give these pump therapies. But we then, amantadine, which you've already mentioned, is a very effective treatment. But amantadine doesn't work on the dopamine system, does it? I mean, so how does amantadine help you when you've got this pulsatile release of dopamine driving the dyskinesia? So what has the work on amantadine told us? And then leading on from that, what are the critical other systems? So obviously you talked about the 5-HT system, the serotonin system. So how does amantadine work, and where else should we be looking for therapies?
I mean, how does amantadine work? My question, sorry. My answer to that is that I'm not sure. I mean, classically, people say amantadine works by antagonizing a type of receptors of a substance called glutamate, so the NMDA receptor.
Yeah.
But amantadine is not a selective drug, so it interacts with nicotinic acetylcholine receptors, and we know that if we activate those receptors over a long time, we're going to get the dyskinesia reduction. It blocks potassium channel. I think it interacts with sigma receptors, so it's not a drug that is selective. And I mean, many studies that have tried to selectively modulate NMDA or similar kind of glutamatergic receptors. I'm not talking about the metabotropic, which is another topic, but the ionotropic glutamate receptors. They have actually not done that well. So I do believe that there is something with amantadine that is probably beyond glutamate. What is it exactly?
And so just to clarify that, so if we just assume it's glutamate, presumably the assumption is that the glutamate is in some way driving a pathway abnormality in the dopamine signaling, and you're trying to block it with amantadine because the dopamine system's gone, or is it working on the glutamate input to the dopamine terminals or the glutamate input to where dopamine works? So where is, I mean, you've already said that amantadine, we're not quite sure how it works and where it works, but I mean, would that be, so I'm trying to work out if amantadine works on the glutamate receptor, where is that glutamate receptor? Is that in the striatum? Is that on the cells where dopamine normally works? Is it on the remaining dopamine terminals? Where do we think it works?
You know, I mean, over the years, I've become a bit cynical, just because, I mean, we think, so the classic explanation is that the reduction of dopamine in the striatum leads to an excess of glutamate in the striatum, which actually drives the dyskinetic state. And if you block or dampen the glutamatergic transmission, then you're going to alleviate dyskinesias. But the thing is that, okay, but my point is, what about the other brain structures? Like, we've been testing a new mechanism of action, which is related to the glycinergic system with GlyT1 inhibitors, which actually should increase NMDA effect. And we obtained a reduction.
They associate with the NMDA receptor and modulate its function. Is that the?
That is correct.
Yeah.
And essentially, based on the conventional understanding of dyskinesias, we should have had the worsening of dyskinesias, but that's not what we got.
Yeah.
And I think that it's because there's many brain areas that just make it very difficult to know where a drug exactly elicit its effect. And it's possible sometimes, I'm wondering if the fact that some drugs don't work to their full potential is that even if it makes sense biologically, if we try to explain the mechanism of action of a drug by an effect at a single synapse, well, what happens in the other parts of the brain? And maybe that can just nullify this hypothetical effect. Or so I find it's very difficult to know. That being said, and this is something that animal models enable to do, and it's been done in the past. So if we really want to explore what is the effect of a given structure on dyskinesias, for instance, so studies have inserted probes to inject a substance specifically in an area of interest and show that it may or not work, which is super interesting from and important also to refine our understanding of dyskinesias, because drugs are being developed that may bind to selective populations of receptors in certain parts of the brain. So it's not so much science fiction at this stage. So it's really exciting.
Okay, so basically we're not quite sure how these drugs work, but we think there's some sort of complex interaction between downstream of where dopamine works and the structures which are innervating or coming into the striatum. So there are a couple of questions here which I think about coming back to an earlier point. If we could deliver, we had this problem of delivering dopamine continuously with a pump, trying to do it in the brain. Could we have a continuous dopamine delivery in the brain, which would circumvent all these other problems, which you say, even if you deliver it peripherally, it changes with food? So is there any evidence that you can do this by putting some central dopamine delivery system into the brain? That's one question. And the second is coming back to this point about manipulating the system. Someone's asked, if it's glutamate that's so important and you cut the corticostriatal pathway, which is glutamate, into the striatum, do you get rid of dyskinesias? Because in your animal models, if you cut the cortical input to the striatum, does that get rid of dyskinesias?
Okay, so maybe onto the first point. So the continuous dopamine delivery. I mean, the problem with every surgical approach, and that would involve a surgical approach, is that it can be very seducing, but it's risky. There are risks during the procedure, and we're talking about there can be bleeding, there can be infection, there can be malfunction. And if, for instance, the device is inside the brain and stops functioning, well, what do you do with that? And then they would probably need to be connected to some sorts of tube with dopamine. Where would that go? So these are all sorts of issues that need to be addressed. But I would say the first thing is, if you have a condition which is Parkinson's disease that can be well controlled for many years with a pill, is it worth exposing someone to a potentially risky surgical procedure that may not yield the benefits that we're expecting? Because there's a danger. From an ethical perspective, I think I think it needs to be discussed because we are exposing patients to risks that would otherwise not happen with a pill. And it's possible that in a few years, maybe dyskinesias will be cured. I mean, I'm hopeful.
Let's go there. I mean, Adrian's very kindly put a link up here to people who are working on this. And obviously, we put dopamine cells in people's brains on that sort of assumption. People have done dopamine gene therapies, and there was even polymers I remember done 30 years ago, which didn't really work because you couldn't get exactly as you're saying. I mean, you put a polymer and it would slowly release dopamine. These were in animal models, but you had to replace it at some point, and that was very traumatic. So I think the idea that you can control dyskinesias by continuous delivery of dopamine has some merit, but clearly, as you've said, that's not going to solve the problem. It's not going to solve the problem in the near future. So where should we be looking? Where's your research taking us? So we've heard about amantadine, and we don't quite know how it works. We've been hearing about your sort of manipulation of glycine receptors, giving you somewhat paradoxical results. So where else are people looking to try and better treat dyskinesias? You know, what is coming on? Because some patients, for example, tell me, and they would have to remain anonymous, that, you know, if they have a little bit of weed, so have a little bit of cannabis, that makes them feel much better with respect to dyskinesias, and that could be because they're just more relaxed and less anxious, which obviously can drive the dyskinesias. But there's a lot of interest in, you know, the cannabis receptors, the cannabinoid receptors. So what are the other pathways which people are exploring around dyskinesias, and what is the sort of hope going forward where we might find new treatments for this to help people with this problem?
So as you may know, well, I know that you had Adrian, and I think he's online a few sessions ago. So there's a lot of hope about 5-HT1A receptors. I sincerely hope it's going to work. I mean, fingers crossed. In the past, there has also been a lot of studies about serotonin 2A receptors, which led to the approval of pimavanserin, but for the treatment of psychosis. But there's a lot of preclinical data showing that antagonizing serotonin 2A receptors might also be a very good way to alleviate dyskinesias and without interfering with the anti-Parkinsonian action of Levodopa. My group has been working on glutamate. So we're working on metabotropic glutamate receptors type 2. and we've been working on the glycine system. So we've been working on these two targets for almost a decade now, and we do have some interesting results. There are some studies with, believe it or not, so ketamine,
Oh, yeah,
which is also NMDA. So we're still very much in the glutamate field. There's this study with mesdopetam, which is kind of a special. So there's 5 types of dopamine receptors, and this one is a dopamine D3 antagonist. Usually in Parkinson's, our understanding of the disease is mostly around dopamine D1 and D2 receptors, but this one is about D3. There's this hype about cannabis. As you mentioned, there had been some early studies 20 years ago that were inconclusive, I could say. There was this compound. I mean, I don't really know where the field is going to go with metabotropic glutamate receptors type 4 and 5. Yeah. There has been a recent study with an mGlu4 positive allosteric modulator that was unfortunate.
So these are all targeting different bits of the glutamate sort of receptors. So they're very
Exactly.
complicated, glutamate receptor. And so the idea is to modulate different elements of it without blocking it or switching it on or off. You're modulating it, and that should help mediate some of these benefits. Is that the sort of idea?
Well, that's it. And there was also a compound called dipraglurant that was in phase 2b/3 trial, so like late clinical testing for dyskinesias. But unfortunately, they had difficulty recruiting, and the development of that molecule, for dyskinesia endpoints at least, is halted. Who knows what the future will be? So there is a lot of stuff happening. I have to say, there's a paucity of mechanism of action. It's mostly centered around a few glutamate and a few serotonin receptors. But I mean, maybe that's the way to go.
Yeah. I mean, it's a great unmet need, as we've agreed. I mean, I think in clinic, as you say, you see most people will develop dyskinesia at some point, and many people, they're not particularly troublesome. Often in the early stages, people don't even notice them. But once you start to move towards troublesome ones, you use amantadine, and if that doesn't work, then you are left thinking deep brain stimulation, all these pump therapies, apomorphine, duodopa, these type of things. So it is difficult to treat. So I think your work and those of others is absolutely wonderful, because you're taking on a problem for which we don't have very much. And I think this idea of probing this, with Adrian and others, the 5-HT system seems very promising. The glutamate system seems very promising. But my sort of final question is, do you think, because people always say, in the next 5 years, do you think in the next 5 years we will have a new drug to offer people for dyskinesias? Do you feel optimistic that the drugs have developed to the point where we think that is likely to happen, or do you think not?
I think 5 years is short. I mean, if you were to, just because we know how long it takes to conduct clinical trials and then the approval process and all that, if you asked me the question 10 years from now, I would say yes. 5 years from now, I would say, I hope so.
Okay. Well, our time is up, Philippe. I want to thank you very much for your fascinating story about how you were determined to be a neurologist or psychiatrist from the get-go, how you pursued that, how you've taken on a topic which has been an area of great interest, but have attacked it with great passion and a lot of innovation. And we're very optimistic that this will yield new therapies. So I want to thank you very much for a very interesting discussion, also around animal modeling, which I think is a very important area which we often don't discuss. And I think it's very useful to hear how that is done and how helpful that is for the clinical translation. So
thank you so much.
Very much for your conversation tonight.
Thank you.
I would like to thank our audience for all of their questions. I'm sorry there's a lot of interest in Mucuna, I'll tell you that. And perhaps we can make that a topic of another research spotlight. But but thank you all very much. I think our next one of these, Eli, is in September with Jeff Corder from Arizona. So thank you all very much for joining us. And for those who joined late and missed the beginning, you can catch up on various different links that we will send you. So thank you all very much, and thank you again, Philippe, for a fascinating conversation.